And it is my pleasure to introduce Dr. Sandeep Gopalakrishan, Director, Wound Healing and Tissue Repair Laboratory, University of Wisconsin, Milwaukee.
Yeah, sure. I'll try to share my screen. Okay, the topic for my discussion today is oxidative stress, the holy grail of wound healing. So my current program of research is technically multifaceted and interdisciplinary. For the past 3-4 years, I have been working with big data analytics as well. So I am a co-investigator in a big data analytics lab, developing newer models for using AI, machine learning platforms to predict the healing characteristics of wound, especially the wound using the clinical data. So that the clinical data can be wound images, electronic health records, and we even play around with synthetic data to develop a diagnostic as well as prognostic tools. So that is some background regarding my current area of research. And another focal area that I have is basically understanding the mechanisms of chronic non-healing wounds. So the title, as you can see from the presentation today, is Oxidative Stress, the Holy Grail of Wound Healing. The reason why I picked this topic is because I felt it is really appropriate for a biochemistry seminar where I don't have to explain too much regarding the free radical theory or anything associated with the free radical associated physiological as well as pathological states. And obviously, to this crowd, I don't think I have to explain all these fine characteristics. So it's easy for me as a presenter to basically navigate through the slides moving forward. So what are some of the plans that we have today? So we'll be discussing some fundamentals to bring everyone to the same page. Basically discussing some of the historical perspective of wounds. And we all know that skin is a multitasking organ. So understanding some fine details of that as well. I will be describing some of the phases associated with wound healing, the molecular characteristics of wounds that doesn't heal, and slowly switch my gears to understand the cellular features in the context of reactive oxygen species and its role in wound healing. And again, the idea of reactive oxygen species and its role in chronic diseases is not novel. And again, it's not novel in the wound healing world as well. So from there, we slowly will switch our gears to understand oxidative stress-associated inflammatory components, and is exactly these contributors have any role to play in the chronicity associated with wound healing. Then a very brief discussion about some of the antioxidant protocols or antioxidant targets that we can have based upon the free radical mechanisms. My lab has done extensive work on something known as redox imaging, which basically works around the domain of free radical mechanisms and oxidative stress. So I will share some results from redox imaging, especially with the wound healing. At the same time, I will share some results using another animal model of neurodegeneration as well, which is known as retinitis pigmentosa. As an intro to the structural characteristics of the skin, as you all know, people wear their health on their skin, right? As the body's largest organ, skin is one of the first line of defense against infections and injury. It is a thermoregulatory organ as well, and it plays a significant role in the endocrine function as well with the vitamin production. So there are sensory capabilities to it. There are feelers that the skin has that basically interacts with the environment all the time. And one pros, you can take it any way you want, but the skin is also very visible, right? So whatever that is being present on the skin is really visible. So it is it basically telegraphs the information to whoever that you are associated with. So most of the time, skin conditions are detrimental psychological effects, and there are newer studies that are coming out which basically correlates anxiety, depression, social isolation in wound healing world as well. These studies have been extensively done during the pandemic period, especially when social isolation was really significant. So think about when you're having a large pressure ulcer, or when you're having a large diabetic foot ulcer in your lower extremity. Probably you can't go for a wedding, you can't go for a social gathering, you can't go for birthdays, you can't interact with people, right? So it becomes a psychological issue at some point of time, and that by itself can be driver of delayed healing process as well. So when you are having stressors in place and stress associated altered characteristics can delay healing further. And one of my PhD students are investigating the mechanisms associated with social isolation and delayed wound healing. So it's a very fascinating area, a newer area. Nothing has been done too much in that field. So if you PubMed, you can see possibly like publications that you can count in your fingertips. So those areas are really promising. Probably you can intervene with psychological effects, or you can have various other psychosocial support system in place that can elaborate or help with the healing process. And to be honest, like most skin diseases are not life-threatening, but most of them comes with some kind of disabilities. When you are having psoriasis, or when you are having these kind of chronic non-healing wounds, it technically don't immediately cause any life-threatening situations, but at the same time, they socially isolate you and can be a cause of a significant disability. And the diagram that you are seeing on the panel left here is probably you would have seen it multiple times during the biology classes, so on and so forth. It basically is the schematic of the skin, which basically involves the full thickness skin locations. So you can see the epidermis, dermis, and various other accessories in that space as well. And on the right panel, what you are seeing is the really significant interfaces between various compartments in the epidermis. And the reason why I picked epidermis is basically because of the fact that most of the initial insults on the skin surface is happening in the epidermis. And there was a recent publication that was basically characterizing the barrier functions at these different layers. So these are all cryo-EM pictures, and those who are familiar with cryo-EM, you can see the barrier characteristics in contrast with various layers. So you can have the granular layer, the lamellar layer, so on and so forth, are really characterized in the epidermis here. And the reason why I'm showing this is to basically show the intact features of the barriers that are present in the epidermis. So these are the ones that gets breached. For example, when you are having a laceration, or when a dog bites you, or when someone scratches you, these are the barriers that gets easily compromised. And understanding the fundamental characteristics of this barrier is important to know what happens when insults happen. At the same time, what are some of the key mechanisms that should be considered when you are thinking about the healing processes as well. So we all age, and it is an inevitable process. So, and you can easily compare your skin 10 years back to now to 10 years later if you're living. So these are all some of the key attributes that are having changes with time. So the complex relationship between the skin at the same time with the kind of age-related changes that are going to accumulate within the skin surface as well. So aged skin is altered structurally, as we all know. Probably if you are having like grandparents or elderly parents at home, you can easily relate this to the fragile nature of the skin, which basically builds in a lot of inherent insult characteristics to the skin profile as well. So with aging, you can see the ECM characteristics are a little bit disorganized. The strength and resilience and the migratory capabilities of the in-house resident inflammatory cells. I don't want to call it inflammatory. The in-house resident infiltrating immune cells will be compromised as well. And as you age, there are cells that are senescent by its characteristics. So fibroblast becomes more senescent, and your capabilities to regenerate decreases with time. So this is an inevitable fate we all will be passing through as we age. So it is a good comparison to make. For example, when you had a wound as a kid versus as a teenager versus now versus as you age, the players there are different. The kind of cellular interactions at the same time, the kind of microenvironment that we are trying to deal here or discuss here are all different. So the intervention for an 80-year-old wound will not be the same for a 50-year-old individual's wound, will not be same for a 10-year-old individual's wound. So all the characteristics are different, the interventions are different, the microenvironments are different as well. So the reason I'm putting these together is to bring a perspective of some of the key regulators and key players within the skin compartment. So the title was the question or the query regarding the Holy Grail, right? So those who are familiar about the Celtic and the Norman legends, many of you would have heard about the King Fisher, and he is also known as the Wounded King. And he is basically, and he has a non-healing wound, okay? He has a perpetual non-healing wound, and his wound starts healing only when he gets replies from one of his mighty knights. His name is Perceval, and those who haven't heard this can go back and refer to it. So the question will be, who does the Grail serve, or what is the whole purpose of this Grail? And as the results comes, or as the knights start responding to these questions, it's similar to the Vikram Vedal story that we had in our mythical platforms as well. So as he start replying in a very, very cognizant manner, the wound starts healing. But the reality is that in tissue repair and wound healing world, many of us follows the line of the Perceval. So you are basically asking a quick question, or a question that is very very tunnel vision, in such a way that you get responses, but that response is not an overall scheme of things, and we are all blinded by the temptation of that simple single answer regarding that particular question that is being asked. For example, even in my research, I will be sometimes blindfolded moving forward, but the biological reality is not the same. That's what I was trying to bring my point to. And the problem of wound healing is nothing new. Everyone knew how to take wounds from time immemorial. Like it has a huge historical perspective as well. For example, when people went to wars, when people fought against each other, when you had animal attacks, so on and so forth. The story of civilization goes hand in hand with a story of injury, war, so on and so forth. So treatment of war is nothing new. It is an ancient art. When I say treatment of war, the treatment of war wounds especially is nothing new. So, and again, these processes have constantly evolved with time. So similar to how the weapons technology has evolved. Easy one to pick probably will be the antiseptic practices and the surgical techniques as well. So an ancient warfare, as the warfare technologies are increasingly evolving, the people who are cognizant enough to basically evolve the technologies used for cleaning the wounds and aseptic measures revolving around the wounds and surgical techniques as well. So some historical perspective, which basically informs us all that wounds are nothing new that we are thinking in a very novel fashion. It's being thought through in various layers during our evolutionary history as well. So the perpetual non-healing wound I described with the King Fisher here is the one that is in the modern world. If you move the time scale further to the 20th and 21st century, the chronic non-healing wounds are the ones that we are discussing here, the one that is perpetually not healing. So it's not going to heal whatever interventions that you are trying to practice here. And in the context of chronic non-healing wounds, there are wounds with multifaceted comorbidities. As you can see in the panel here, there are venous leg ulcers and arterial leg ulcers. So the vocabulary basically gives you a feel of what kind of pathology that you are discussing. One is a venous pathology, the other one is an arterial pathology. So both are vascular. So you don't have enough blood supply, or the vascular supply to that particular location is compromised. Most of the time it is on the lower part of your anatomic peninsula, which is the lower extremities. So ischemia, the lack of blood flow, lack of oxygen to that side. So whenever you are having any abrasions or lacerations in those sites because of the lack of blood flow, it's not going to heal. And a very commonly seen non-healing wound in the context of both US as well as in Indian clinical domain is diabetic foot ulcers, because you have millions of people getting diagnosed with diabetes every year, and diabetic foot ulcer comes as a unique complication with time. So unmanaged or improper hyperglycemia is the fundamental reason behind it. And another class of wounds that probably you are familiar is also the bed sores or pressure sores that you want to call. And again, it is mostly seen in the elderly world or individuals who are mostly mobile. So you need to have a surface that is exerting pressure to your skin surface, and that's because of the comorbid issues. The structural characteristics of the skin is becoming fragile, mostly as you age, mostly as you are immobile for an extended period of time. And these wounds can heal. And interestingly, there are ways in which wound healing happens that are abnormal as well. So you can see two examples here, which are hypertrophic scars as well as keloids. So these are tissue compartments that have regenerative capacity, but the capacity is a little bit exaggerated. So they are healing in a much more proliferative way. Thereby, you end up having excessive growth in that sites as well. So hypertrophic scar, keloids, if you compare that with the wound healing trajectory, they have an abnormal cellular features associated with it. But interestingly, they heal though. So these pathological scarring mechanisms are pathological only because of the fact that they have overhealed that particular site, or they have exaggerated the healing process. So these kind of dermatological findings are commonly seen in individuals who have hyperproliferative profile as well. So this is in the context of chronic non-healing wounds. But in the context of acute wounds, probably we all are familiar about surgical wounds, burn wounds. Burn wounds can be sometimes chronic, depending upon your background, but most of the time they are acute. So post-operative wounds, as we all know, are acute. Then sometimes hematological abnormalities similar to that of sickle cell anemia can cause hypoxia-driven or ischemia-driven wound profiles as well, which are known as the sickle cell ulcers. So these are some of the commonly seen or commonly identified clinical wounds in the dermatology domain. So why is it significant? Why should we discuss about chronic wounds? What is the fundamental reason for discussing these particular issues? One is humanistic, because you are basically compromising the quality of life in these individuals, right? As I have mentioned earlier, when you are having a large a non-healing malodorous wound in your extremities, you have difficulty moving around. You have difficulty interacting socially. You have difficulty having a good quality of life. So in that particular context, it has humanistic perspective, right? So it basically is preventing you to have a good quality of life. And apart from that, on top of that, you have a huge economic burden as well. Think about all these people who are coming for dressing changes, who need to be addressed in the context of very frequent intervals in wound care clinics or in dermatology clinics. So I have pulled some statistics here. So in US, it is a huge issue. As you all know, there is a huge aging population here, which brings in fragile characteristics to the skin. Thereby, you end up having chronic wounds. Not only the increasing aging population, but having a larger diabetic population as well. So rough estimates basically say that you have a cost of close to $50 billion in healthcare spending and expenditure just in the context of chronic non-healing wounds. And Indian statistics is also not far from this. So we have a we have an increasing burden with newly diagnosed diabetic population as well. And India ranks second, if I am correct. I think China goes first, and India ranks second in the number of diabetic individuals as well. So of the newly diagnosed individuals, close to like 25% of them ends up having diabetic foot ulcers. They may get infected. They may require recurrent hospital admissions and hospitalizations. Some may even go to amputations. And the Indian statistics basically shows that of the 25% who develop DFUs, 20% requires amputation. Think about the quality that gets compromised there, and think about the disability-related issues that those individuals have to deal with as well. And DFUs contribute approximately 80% of all non-traumatic amputation. That's a huge number. So those numbers come in like probably hundreds of thousands, and not only that, we don't have enough resources to cater these individuals who require amputations. At the same time, it brings a personal economic burden to those families. At the same time, it's an economic burden to the healthcare systems as well. So one of my friends, Dave Armstrong, who is at University of Southern California, basically compares chronic wounds with the cancer statistics, and he used to equate the DFU mortality or diabetic. And again, this is just by diabetic foot ulcers. Okay, I'm not even considering other chronic non-healing wounds. Just by picking diabetic foot ulcers, the mortality is very close to that of the all cancer mortalities, as you can see here, as a 5-year mortality rate. So, and again, you can see chronic limb ischemic features over there, major amputations over there, so on and so forth. So by considering the clinical magnitude, we care. So when you hear that an individual is diagnosed with cancer, so there is a clinical magnitude to it, right? A personal magnitude to that particular vocabulary when you hear that, oh, that individual got diagnosed with cancer. But that doesn't goes very well with the vocabulary of a foot sore or a foot ulcer. But this can be very well equated in the I know it is a little cheeky to compare DFU to cancer, but at the same time, we need to compare and contrast it with a significant problem clinically in such a way that you start addressing diabetic foot ulcers or chronic non-healing wounds in a similar category, so that the outcomes in patient populations becomes better. So the cancer analogy sometimes becomes powerful. So powerful in the sense for communicating the risk to organizing care for the patients with this kind of diabetic foot syndrome, or I am picking diabetic foot syndrome because the statistics are on that, but chronic non-healing foot syndrome as well. And again, one potentially underappreciated similarity between cancer and foot ulcer is that both can recur. So for example, if I am having a diabetic foot ulcer and if it goes to remission, at some point of time, it can come back, correct? At some point of time, it can come back. So the clinical trajectory is very similar to that of cancer clinical trajectory. So you have a particular cancer diagnosis, you treat for it, you manage for it, the cancer goes off, so you become cancer-free for X amount of time. But after some time, it can come back, right? It can come back, so it can recur. And interestingly, this can happen with diabetic foot ulcers or chronic non-healing wounds as well. So you may have a particular wound in a particular anatomical location. You took care of it really well. You control your diabetic profile really well. Yeah, you managed it really well. You had resources, and when I say that, you should be always cognizant about the fact that you had resources around to manage that as well. So you managed that really well in such a way that the wound healed. But the problem here is that it can recur at a different anatomical location, and it can be distinct from that primary occurrence as well, even though the physiological mechanisms can be albeit. So the point here is that the recurrence of these sores or these wounds in a different anatomical location goes hand in hand with the discussion that we have or that we are aware regarding the cancer biology. So that's a very good comparison to make, because as you start making that comparison, you start become much more cognizant about some of the key clinical landmarks that these issues can bring down with time. I will pause a moment here. I would like to check if there are any questions so far, or am I free to move forward? Any questions, any thoughts?
We can have a discussion at the end.
Okay, that's fine. So moving forward, those who work with the wound healing world knows some of the key physiological mechanisms associated with wound healing. So this is a graphical representation of the important phases associated with wound healing. When I say important phases, there are 4 phases involved in wound healing. You can easily compare with the wounds that you had in your body as well. So when you had a laceration, so you have bleeding from that side. At some point of time, in few minutes, it basically starts not bleeding from that particular site, right? So you have hemostasis in place. Then you have an inflammatory process that goes for an extended period of time. Then you have a proliferative phase, because you have damaged and you need to replace cell populations that you have taken away from that particular site, right? So think about scooping an ice cream from a basket. So when you are scooping an ice cream from a basket, you need to replace that ice cream to make it look as if it is normal, or look as if it is similar to that initial positioning of that. So that happens, and it goes hand in hand with the remodeling as well. So as you can see, the wound healing is a very complex and dynamic physiological process. It requires some spatial as well as temporal synchronization as well. If the temporal synchronization is not there, you end up having keloids, right? The one that I showed you before, because the spatial and temporal synchronization should be there, which basically says that if the volume is V, that needs to be filled. So the proliferation and remodeling should end up filling that space V. It should not be 2V, 3V, or V plus 2, V plus 3. It should not be any other variables of V, correct? So the spatial and temporal synchronization is really important. And a variety of cell populations play distinct roles throughout these phases as well. So the key players of in hemostasis are different from inflammation, and they are different from proliferation, and they are different from remodeling as well. So in the right panel over here, you can see some of the key cellular players. As you can see, the first response to wound is basically, as I have earlier mentioned, when you have seen your wounds, the first response to a wound is a constriction of the injured blood vessel, right? And activation of the platelets to form the insoluble fibrin network. Probably you can refresh this back to the coagulation cascade, so on and so forth. So the goal is to provide, or the goal is basically to stop the bleeding immediately. And then there are a lot of players that are coming there slowly. And again, neutrophils are not a resident cell population in the skin. But as soon as you have injury, you probably would have heard about extravasation of neutrophils from vasculature, right? So neutrophils are immediately recruited from the nearby vasculature to the site of clot. And clot basically, clot by itself, platelets by itself, and injured epithelial cells by itself releases cytokines and various cellular mediators. Thereby, neutrophils can be chemotactically attracted to that particular site. So these are considered as the first line of defense, because when you expose your underlying tissues, you are exposing it to the environment, right? And in the environment, you have friends and foes. So you have bacterial population, you have various other microbes that are in line to access that particular site. So these are the first line defense mechanisms, and a neutrophil does this job extensively by the oxidative burst and using free radicals as well. So that again is a really good discussion that goes hand in hand with the oxidative stress component of our discussion. Then as the innate immune system slowly gives away to the adaptive axis, so the resident monocytes, or the monocytes that are circulating, that gets recruited to this particular site in a tissue active manner, becomes tissue active macrophages. And they basically start surveilling this particular site And apart from the macrophages, and I will give much more detailed elaboration of macrophage population, because they are key players in retaining the chronicity of the wound. So it is a very good discussion to have down the road as well. So the adaptive immune system involves various other players as well. There are Langerhans cells, there are dendritic cells, T cells are there, which are involved in combating self as well as foreign antigens. So as the inflammatory phase, as you can see from the diagram here, as the inflammatory phase slowly ends, you can see angiogenesis coming in, and angiogenesis involve endothelial cell proliferation, not only proliferation, the migration and branching to form the newer blood vessels. So when the newer blood vessels emerge in that particular site, the resident fibroblasts start proliferating extensively, depositing the extracellular matrix. They invade the clot to form the contractile granulation tissue. The fibroblasts start differentiating to form something known as a myofibroblast. They are of the same lineage, but the phenotype is different. So they basically differentiate into myofibroblast, creating the wound barriers and wound margins as well. And the repair of the epidermal layer also involves reconstruction of the skin appendages, right? It's not only the epithelial layer has epithelial cells, it has hair follicles, it has sebaceous glands, it has resident stem cells, especially in the hair follicles, so on and so forth, which need to be repaired as well during this repair activating process. So, and again, nothing has been done too much in that particular domain, especially with skin appendage reconstruction and skin appendage regeneration as well. So that's a whole area, new area. There are a lot of studies that is coming out, especially with the stem cells in that domain. So it is important to understand that our knowledge of skin repair and cellular architecture of healing wounds are mostly generated from animal models, right? We do experiments on animals. We all do experiments on animals, but we never saw a rat running around or a pig running around with a wound, correct? So if you look at wild, if you look at wild animals, or if you look at the wild, you generally don't see animals running around with wounds, because their healing mechanisms. And again, this is the problem with the wound healing world, because the rodent models have been used for from the beginning of the wound healing research. Murine models are extensively used, but they don't exactly compare and contrast with the human situation, right? The closer models are the porcine, but still it doesn't completely establish the impaired wound states that you see with diabetes or aging or various other hemoglobinopathies in the context of sickle cell ulcers that we have discussed. And rodent skin, unlike human or porcine skin, is more elastic. Probably those who work with wound healing can see that the day after inducing the wound becomes a little larger because of the elastic characteristics of the wound, and it lacks adherence to underlying tissues properly. And it basically closes in an entirely different way. So wound closure is happening by contraction stimulated by a striated muscle and panicculus carnosus of the striated muscle. So that is a big problem in the wound care world. Whatever you try to establish with rodent models, one-to-one doesn't translate to human wound healing issues, or it doesn't completely translate to the way human skin heals by granulation tissue formation and reepithelialization. So that's a huge debatable topic within the researchers who work with the wound care domain. So what exactly? So we discussed what is happening with the normal wound healing, right? So these are some of the key parameters that drive normal healing process, but we are not interested in what is happening normally. What we are trying to do is what is happening in a chronic wound. So what are some of the molecular pathologies of chronic wounds? So this illustration basically gives you a feel of that. So chronic wounds show some kind of hyperproliferative characteristics, but interestingly, they have a non-migratory epidermis. They have unresolved inflammation. So it is wise to say that the chronic wounds are stalled in the inflammatory. If you go back again to our phases of normal wound healing, it is wise to say that chronic non-healing wounds are stalled at the inflammatory phase. And why is it stalled at the inflammatory phase? That's a very interesting question. Oxidative stress has a role to play it, but there are tons of other players that are stalling inflammatory stage or stalling inflammatory phase in the chronic non-healing wounds. So there are hyperproliferative characteristics to the wound bed, but there are non-migratory epidermis that can migrate and can fill the space of the void that has been created. There are unresolved inflammation. I already mentioned that. And again, a very fascinating component here is the presence of infections and biofilm formations. So I will discuss one of my collaborators' work who basically establishes that not only oxidative stress, but biofilms by bacterial generation is required for the chronicity of a particular chronic wound. Although there is an increase in the inflammatory cells, not all are properly functioning, as you can see. There are a lot of senescent cells that are not capable of doing the particular function as well. So, and again, depending upon what you are discussing, whether it's a diabetic foot ulcer or a pressure ulcer or a venous leg ulcer, the kind of dysregulation in a molecular level is a little different. So this will give you an overall picture of what are some of the key players involved in this particular process. And this is an extension of what we have discussed earlier. So I would like to bring your attention to some of the key cellular players that are trafficking these sites during different stages of healing. So you can see during during the hemostasis part of the wound healing, you have a particular cell population. During the early versus late inflammation, you have a particular cell population. And during the resolution remodeling phase, you have different cell populations as well. So being cognizant about these cell populations that are trafficking these sites are really important in the context of understanding the healing trajectory as well. So ROS plays a significant role, and its role in wound healing can be linked to almost every phases of wound healing that we have already discussed. So ROS is critical for hemostasis as well as establishing the overall homeostasis in the site as well. So we discussed the fact that ROS-mediated vasoconstriction is significantly important to drive hemostasis. At the same time, you have various kinds of inflammatory cell recruitment, not only the neutrophils, but the cells of the adaptive immune system as well. The T cells, the B cells, as well as the macrophages are key cells that is being recruited to that particular site to achieve various functions that we have discussed. ROS drives tissue repair as well because they mediate cell division, migration, and proliferation of cells that need to be replaced. For example, keratinocytes, endothelial cells, fibroblasts, so on and so forth. So as they get replaced, then the ECM matrix will be synthesized as well. So tissue repair, ROS plays a significant role. And in an overall scheme of thing, ROS has a role to play in multifaceted level in the context of wound healing. And this is a different version of different reactive oxygen species and its components involved in wound healing processes as well. So in the thrombogenic cycle of initial hemostatic cycles, we have significant free radical players. So I'm not going into the free radical machinery here because I am pretty much aware that in a biochemistry colloquium, everyone is at least aware about the fact that free radical, what are free radicals, what are some of the key regulatory components that drive free radical generation as well. So the site gets disinfected because of free radicals. It allows for chemotaxis. The different cell players are brought into that site. It helps with reepithelialization. It helps with revascularization, so on and so forth. So in an overall scheme of things, ROS has a huge role to play. And as you can see, there are different molecular as well as cellular deficiencies that we have already discussed. And as you can see, many of them revolves around the ROS-mediated mechanism. So inflammation revolves around ROS-mediated mechanism. inhibition of proteases revolves around ROS-mediated mechanism. The altered growth factors and ECM deposition revolves around ROS-mediated mechanism. So ROS has a huge role. When I say ROS, the reason why I'm picking that particular vocabulary is to basically say that you can start modulating that, right? So as you know that you can target a particular molecule, you can start modulating that particular molecule as well. So there are tons of literature evidence where you can start modulating the ROS in the wound bed in such a way that you end up having a better outcome. When I say better outcome, the outcomes in the context of having an accelerated healing. So there are two different kinds of approaches at this point of time. There are recombinant technologies. There are commonly used clinical intervention technologies similar to that of hyperbaric oxygen therapy, through which you enhances the oxygen concentration in the wound bed. Thereby, when you increase the oxygen concentration in the wound bed, technically you are creating free radicals there as well. So there are different ways, different approaches, different targets that you can use to effectively modulate ROS. And as you can see in the table over here, there are outcomes that are beneficial in the context of healing. They are antibacterial. They promote healing characteristics. They allow proliferation to happen. They allow migration to happen. They allow recruitment of immune cells, both innate and adaptive, to allow healing events to happen. They helps fibroblast to change its phenotype to myofibroblast. They help angiogenesis, so on and so forth. So the goal here from this slide is to basically reestablish the fact that the oxidative stress plays a critical role in the initial as well as during the later phases of wound healing. But at the same time, you can be smart enough to target those sites, and by targeting those sites, you can have better outcomes as well. So now I want to link the ROS profile that we have discussed so far to the inflammatory component as well. So inflammation driven by ROS is some of the mechanisms that I will be discussing in the next few slides. So more recent studies have focused on like innate immune sensing receptors and their role in inflammation as well as in disease processes similar to chronic non-healing wounds. And all of us have heard about the NLRP3 inflammasome, which is technically a molecular platform that can modulate innate immune functions by activating series of caspases, and it can cause proteolytic processing and secretion of interleukins, especially the 1 beta as well as 18 from the immune cells, so on and so forth. And this cascade triggers sustained inflammation, right? Those who know the NLRP3 inflammasome mechanism, when you are having increased oxidative stress that basically contributes to an upregulated NLRP3 axis, you have a sustained inflammatory profile. And in chronic non-healing wounds, these NLRP3 upregulation has been demonstrated by multiple literature evidence as well. So the cascade, the ROS cascade that can trigger sustained inflammation is a critical player here, because as we know that this is a critical player, you can target these significant molecules as well. And again, ROS in a specific definition, it can be microenvironmentally triggered at the wound bed, or it can be because of a macroenvironment characteristic as well. So I would like to bring your attention to a study regarding ROS-activated NLRP3 inflammasome, especially with the from here onwards, most of the studies that I will be quoting and I will be discussing is exclusively on the diabetic domain, so diabetic foot ulcers or animal models that recapitulate the diabetic models or the animal models. Even though I previously mentioned that this can't be one-to-one translated, the mechanistic aspect of it can be investigated in a very, very, very cognizant way. And I would like to reemphasize the fact that it should be in a cognizant way, because what you are seeing in the animal model does not always reflect with the human population. So from this particular study, you can see that NLRP3 protein expression is upregulated. Same is true with the caspase, because caspase activation modulates the NLRP3 activity as well. So different kinds of caspases are listed over there. And the goal from this particular manuscript is to basically demonstrate the fact that excessive ROS production, when I say excessive ROS production, then you are moving into the boundary of oxidative stress. So when you have excessive ROS production that your endogenous antioxidant defense systems can't deal with, then you are discussing more towards an oxidative stress, which basically contributes to the inflammasome activation that we have discussed here. And I don't want to go to the fine details of it, just to give you a brief intro that these inflammasome molecules that goes hand in hand with various other interleukin platforms are impaired in wound healing, and they can be one of the targets as we design newer drugs and molecular targets as well. I briefly mentioned about macrophages earlier, correct? I briefly mentioned about macrophages in the context of the resident monocytes becoming macrophages. At the same time, the switch in the macrophage profile from an innate to more adaptive as the wound bed becomes more adaptive by its immune characteristics. So macrophage is a very fascinating cell in the context of wound healing because they play a significant role in driving all the phases of wound healing, especially from the inflammatory to the remodeling phases. So in vivo macrophages are involved in the pro-inflammatory characteristics of the wound. They basically are involved in resolving the inflammatory components as well. So I will tell the switch in the macrophage profile pretty soon. But the take home from these discussions is to basically make us aware about the fact that macrophages are fine players in the wound bed, and the switch in the profile of macrophage is one of the key regulators of chronicity of wound bed. And in the left panel, as you can see, whenever you are having injury, whether it's a burn or a laceration or a cut or whatever it is, or a radiation injury, whatever it is, the epithelial cells that are getting damaged over time will basically release interleukins, TSLP, so on and so forth, that can prime the monocytes to that particular site. So the monocytes will be recruited to that particular site. At the same time, some macrophages will be involved in phagocytic activity as well, right? So they, for example, the neutrophils that have completed their job during the early phases needs to be cleared. So the neutrophils undergo apoptotic pathways, thereby it gets cleared. At the same time, the apoptotic remnants are cleared by the macrophages as well. These resolving macrophages help to even phagocytose the resident neutrophils that has already done its job as well. So the role played by macrophages are really multifaceted here, and switch in the role is also really important here to understand. So how well the switch is happening and what are some of the significant modulators of that switch. And in the next slide, you will be able to see, I will try to bring your attention to the panel on the right, where you have a non-diabetic wound, where you have a unique population of macrophages known as M1 phenotype, which is upregulated there. So the normal phenotype transition happens there, versus in a diabetic wound, you have a much more pro-inflammatory transition that is happening there, which basically persists inflammatory activities within the wound bed, thereby the inflammation is persistent. So that is again a key mechanism to understand in the context of macrophage switching and phenotype transition. On the left panel, what you are seeing here is a potential mechanism through which bone marrow can recruit macrophages, especially in the context of a normal wound healing profile versus one with a diabetic wound healing profile. And again, as we all know, diabetes by itself is providing a systemic low-grade inflammatory characteristic. So that particular low-grade chronic inflammation induces a different kind of myeloid bias in the diabetic hematopoietic stem cells, leading to the increased production of macrophages that are delineated to be differentiated in a particular way. So these intrinsic modifications are also really important, and various other epigenetic modifications. I was discussing epigenetic modifications with Dr. Allen and Dr. Annie before we started our discussion. So these are some of the key epigenetic modifications. For example, hypermethylation happening in various genes. For example, the Notch, KLF, and PU genes are one of the few genes that undergo methylation-related epigenetic modifications as well. So to understand these changes that are happening in the bone marrow compartment, at the same time, increased recruitment or increased level of various kinds of DAMP agents and inflammatory cytokines and chemokines are significant to basically reroute our discussion back to the NLRP3 discussion that we had, NLRP3 and the IL-1 signaling discussion that we had as well. So it's a vicious cycle. So when you start profiling your macrophages in a particular way because of the characteristics of diabetes, because of the characteristics of the chronic non-healing wound, this vicious cycle go back and start recruiting more macrophages to that site, which are bound to delineate into a much more pro-inflammatory phase. So the macrophage phenotypic switch is highly compromised in chronic non-healing wound. So one of my colleagues, Manuela from UC Riverside, recently published a manuscript which basically compares the high levels of oxidative stress along with the skin microbiota, because we can't have independent discussion of oxidative stress in a wound bed, because obviously as soon as you are having a wound, microbial colonization happens. Microbial players drive the rest of the key events that is happening in the wound bed as well. So an interesting take home from this particular slide, as you can see, is that obviously it is very clear that as you are having an increased oxidative stress, the healing is delayed, right? So the chronicity is established by obviously by the characteristics associated with an increased oxidative stress, which is very clear and which is very straightforward here, right? There is a dose-dependent effects of oxidative stress on the levels of wound chronicity, which is not novel by itself, but which is reestablishing the fact that oxidative stress is a key player in establishing the chronicity of the wound bed. So this is again a dose-dependent response to the wound closure. So wound closure is delayed in wounds with the higher oxidative stress, as you can see. And percent wound closure was calculated by measuring, I think if I am correct, I think it was calculated by measuring the area of the wound from the beginning to the 20 days after wounding. So that's how I think they calculated that particular variable. And interestingly, when they stain for HND, when they stain from Mason's trichrome, when they did the picrocyrus red, when they did for the collagen staining, so on and so forth, you can see the characteristic changes that is happening in the wound bed that has higher oxidative stress when compared to that of the lower one. So this brings into the attention that not only the oxidative stress is involved in driving the chronicity, but the driving the chronicity is driven mostly through altering the ECM metrics and altering the characteristics of the ECM metrics as well. So these immunofluorescent stainings, especially the one that the M and P, that the M from M through P that you are seeing over there, the immunofluorescent staining for collagen 4 shows the presence, especially in the basal lamina underneath the epidermis. So that white arrows that you are seeing over there indicates the basal lamina is formed below the basal cells of the epidermis. So this basically calls for an altered structural profiling that is happening, or an altered structural feature that is happening in the context of having a higher oxidative stress as well. This again tells you, brings you a similar story, but what I would like to bring your attention is to basically the lower right panel, those bar diagrams over there. So you can see with the increased oxidative stress, the resident macrophage numbers are increasing. That goes hand in hand with the discussions that we had earlier with the macrophage phenotype and switches that is happening in our earlier discussion. So with that, this particular study is reestablishing the fact that the oxidative stress has a huge role to play in dictating the type of macrophage. They haven't typed the macrophages. That is one critical review that probably I have for this particular manuscript. They would have done the macrophage phenotyping, which they haven't done. They have done the overall number of macrophages that gets recruited to that particular site. So as you can see, it increases with the kind of oxidative stress that you are seeing over there. So the take home from these studies is that they are basically able to demonstrate that the oxidative stress levels in wound tissues are critical for the chronic wound initiation as well as development, and they are able to show that. I took the verbatim from their manuscript. So they were able to show that increased OS levels in the wound correlate with the increased chronicity as well. Then let's bring into the equation the microbial colonization in that particular site. So as you can see, with microbial colonization, the situation becomes much more worse. So based upon the free radical stress, you can see different kind of bacterial population that is getting colocalized in that particular site, and the bacterial profile is being listed there as well. So I would like to have your attention to the right upper panel over there, which basically is discussing in an abstract manner what are the effects on diabetic mice with respect to oxidative stress, as well as what are the effects that skin microbiome is contributing to as well. And so that brings you a very interesting discussion in such a way that both the skin microbiome as well as the free radical characteristics of the oxidative stress are required for the wound chronicity characteristics. And again, one by itself can impair and alter the wound healing characteristics. So for example, having an increased oxidative stress obviously alters the wound healing trajectory, and that is true with having bacterial colonization and biofilm as well. And again, in this particular context, I would like to invite your attention to microbial colonies that basically enhances healing as well. That is there as well. So we should not be forgetting the fact that there are colonies that can enable healing characteristics in the wound. So there are immense studies that is going on to basically characterizes the bacterial colonization that can accelerate the healing process so that they can be utilized as a therapeutic tool as well. So the take home here is that even though the oxidative stress and colonization by itself delays the healing trajectory of a wound, to establish chronicity in a diabetic model of diabetic murine model over here, both is required. So they are basically saying that the synergetic effect of oxidative stress and oxidative stress that goes along with the bacterial colonization is a key player for initiating chronicity in the wound bed. So from here, I will switch gears and will discuss some of the key imaging studies that has been done in my lab, especially the redox imaging studies that has been done. So one of the studies is basically to characterize the mitochondrial, and again, I don't have to go back to a biochemistry audience to basically discuss mitochondrial dysfunction that goes hand in hand with the altered oxidative stress characteristics. So we were able to demonstrate that the mitochondrial redox state is unique with respect to diabetic wounds. So when I say mitochondrial redox state, we are basically looking at the NADH-FAD ratio using non-invasive imaging techniques. As you can see the instrumentation over there, and we basically image the animals in a longitudinal fashion from day 0 to day 6. As you can see on day 0, almost all wounds are having characteristic area and architecture, but as time progresses, the wounds that are in the control animals, which are the ST animals, versus the diabetic animals or the DBDB mice. So in that particular study, we were able to demonstrate the fact that the wound healing trajectory can be predicted by the mitochondrial redox states. And again, using the next step of studies that we are trying to do at this point is to basically use AI machine learning platform to have these kind of images being used as predictive variables for the healing trajectory. So you will be able to see the characteristics switch in the redox ratio with time. So the diabetic wounds are much more altered by its redox characteristics, and mitochondrial dysfunction can be easily tagged to that particular feature as well. So fluorescence imaging basically establishes the altered redox state in these wounds. And then obviously targeting mitochondria is a key intervention that you can do when you start understanding the fact that mitochondrial dysfunction is a key player here. So one of the ways we have targeted mitochondria is by a non-invasive process known as photobiomodulation, where you start modulating the physiological effects of cells by using different wavelengths of light. And the wavelengths of light that we have used here are the far red to near infrared light. So targeting the mitochondria basically allows a better mitochondrial profile. So basically, it improves the mitochondrial redox state of the diabetic wound. So which basically says that the dysregulated mitochondria that results in an increased oxidative stress or an altered redox state can be fixed by intervention similar to that of photobiomodulation. And photobiomodulation is performed either by lasers or by LED light sources at a particular intensity. And you can see the healing trajectory as well as healing characteristics of photobiomodulated tissue there as well. So the faster the healing, better the redox state, you end up having a much more targeted response with wound healing rates as well. So one of my colleagues, Praveen at the University of Buffalo, has done similar studies using photobiomodulation. It is not only us who has demonstrated that. We have other groups that has been working in parallel. They have published their results as well. In the wound healing photobiomodulation world, we were able to publish our work with scientific reports, and it was very well accepted in the field as well. So I would like to bring your attention to a different wound model here. This is a burn wound model, which can be compared to that of an acute wound model. Not always necessarily, but you can compare that. And again, this particular study was able to establish a similar trajectory that we have demonstrated as well. And this particular trajectory basically establishes the fact that the healing can be significantly improved. And they have gone one step further as well. They have gone one step further to basically characterize the signaling pathways associated with it. And TGF-beta signaling is a signaling pathway that has been already established in photobiomodulation studies earlier. So his group was able to basically bring our attention to this signaling pathway again and reestablish the fact that photobiomodulation can activate TGF-beta signaling that can promote wound healing, especially through the epithelial migration characteristics of the wound healing. So I mentioned the fact that apart from the wound healing, we were able to establish a similar imaging protocols in similar other models as well. So retinitis pigmentosa is a unique model of inherited retinal degeneration. It is done using a transgenic animal model known as P23H rats. And a similar characteristics were able to be demonstrated here as well. So structurally, functionally, we were able to characterize mitochondrial redox state. And this is a little bit apart from the wound healing work. But the goal showing this particular work is to basically reestablish the fact that targeting redox alterations in pathologies can be one of the key ways that you can move forward to establish normalcy in those, or I don't want to call it establish normalcy, but establish to at least slow down the process of dysfunctionality in those chronic wound models, as well as in neurodegenerative models as well. One of our work got published as the cover page for Journal of Biomedical Optics as well. So just out of curiosity, I thought of sharing this information. So some of the future studies aligns very well with targeting mitochondria again, targeting mitochondria with the hope of altering the oxidative stress characteristics of the wound bed. So I am working with an investigator from the Salk Institute by the name Pamela, who is basically the individual who discovered and patented a unique molecule called J147. And J147 is a molecule that targets ATP synthase. So by that, we basically submitted an NIH grant proposal a few weeks back. And the pathway that we are trying to envision to investigate is being listed over there. So having ATP synthase being targeted over there will be able to characterize. So the ATP obviously will get screwed up here. So you end up having an altered CAM-KMPK signaling happening, which can downregulate the mTORC pathway, and which can alter the cellular senescent characteristics of the wound bed as well. And to your attention, this is being a study proposed in an impaired wound healing in an aging model, because aging model is much more in align with J147 studies, because J147 is investigated as a neuromodulator in Alzheimer's models in preclinical trials, and currently it's going clinical trials in the United States as well. So to be much more in align with the existing literature, we picked to align ourselves with the aging impaired wound healing model as well. And there are preliminary investigations that we have completed using inhibitors of pheroptosis in various neuronal models, which we were able to demonstrate that free radical characteristics are key in driving these pathologies. So we hope to get this grant funded and move these studies forward as well. And with that, I would like to bring your attention to one more topic before I wind up here. So the recent publications have demonstrated a significant overlap between cancer metastasis as well as non-healing wounds. So that is another area that my lab is trying to focus at this point of time as well. So what are some of the key drivers that helps in playing a unique role in an overlapping feature within a cancer metastatic event as well as a chronic non-healing wound? So the wound healing response involves, as we have seen, interplay between diverse cell populations, right? And these diverse cell populations are the exact ones that are involved in the progression of cancer metastasis as well. So my next goals will be to basically characterize some of those cell populations, and I want to tie this to the wound healing characteristics. So the kind of metastatic profile that you are seeing in these wound bed, in this cancer metastatic event should be which should be tied back to the wound bed. Thereby, you can reestablish some of the key overlapping signaling pathways in these mechanisms as well. With that, I would like to wind up by mentioning a few of my concluding remarks here. Wound healing, as we have seen, is a very complex and dynamic physiological process. In addition to the positive influence ROS provides with various phases of wound healing, excessive ROS is detrimental, and that is true in various other pathological situations as well. And targeting, and similar to J147, photobiomodulation, so on and so forth, targeting these redox alterations are probably future directions to basically alleviate some of the chronicities associated with these disorders. And with that, I would like to share my collaborators. I have collaborators at Salk. I have friends and colleagues at my own institute, as well as at the Florida Atlantic University. And I would like to thank all the lab members and grad students in my lab for doing all the work and allowing to get funds and move the lab forward as well. Yeah, the social isolation aspect of wound healing is really, really new. There is nothing much out there. So especially in Indian context too, identifying, because I personally think that it's a vicious cycle, because the social isolation altering stress responses, and again, there are tons of literature evidence that basically correlates social isolation with anxiety, depression, and various other psychosocial and psychiatric alterations. So that can by itself being a driver for impaired wound healing as well, because these psychiatric issues can alter a variety of your normal characters. For example, it alters your sleep characteristics. Sleep can affect inflammatory responses. Inflammatory responses systemically affected by sleep can go back and locally affect the wound bed as well. So that is the focal topic of research by one of my PhD students.
Sir, which one is the best animal model you are suggesting?
Oh, for the wound healing, the best animal model to play around is the porcine model, but it's very expensive, right? The porcine models are relatively expensive when compared to that of the rodent models. I have problem with rodent models all the time because if you observe the next few days after the induction of wounds, you can see all kind of profiles in the rodent model because the skin is really elastic. But the porcine models are the most accepted ones. But the problem is that it's too expensive and not all animal facilities are able to deal with that.
MicroRNAs are being therapeutically, wound healing can be improved. So
Correct.
have you done any experiments related to microRNAs?
Yeah, not only microRNAs, small microRNAs are also key players in wound healing now. One of my mentors in this field, Chandan Sen at the Indiana University, is a key player in that field, and he has done a lot of microRNA studies on wound healing as well. Not only as I have mentioned, not only the microRNA by itself, but the small microRNAs are also being shown to, because as soon as you start modulating the microRNA profile through which you can characterize different expressions by the cell, you can change the fate of the cell more towards a healing trajectory when compared to that of having it stalled in the inflammatory phase. Yeah, there are a lot of studies going on with microRNA, and those are newer areas, probably more therapeutic by itself, because you know that microRNA technologies are used now for vaccines, used now for various other clinical requirements. So probably down the road, microRNA mediated targeting can be one of the ways that you can reestablish normalcy in the chronic wound bed. And again, one good thing with the chronic wound bed is that most of the time it is localized, right? So when you start playing around with the local wound microenvironment, you have much more better outcomes when compared to when you start playing systematically, because you never know what are some of its other consequences in other organelles and system sites.
One question, probably you might have also experienced this from your own observation. I was saying that all these Chinese people, they have particularly younger skin, even though they are very old. So in the starting, you said that aging process, skin is an inevitable process. What is their difference from us? Is it genetic or is it some other?
Yeah, there are racial differentiation in the extra ECM composition as well. So a 50-year-old Chinese guy probably doesn't look like a 50-year-old Indian or Southeast Asian or any other geographic. That is true as well. But I don't think there are mechanistic investigations that is done regarding that particular question. But probably the fullness of the skin comes from the ECM that basically holds the different layers, right? So as soon as it starts disintegrating and as soon as it starts having much more frail characteristics to it, it starts crumbling, and it starts crumbling. It starts having the wrinkled characteristics to it. So probably I haven't come across any mechanistic studies investigating it, but I have also observed the same. We have even told many times between ourselves that, oh, look at that guy in the Chinese restaurant. He doesn't look that old.
I always had a difficulty gauging their age.
Yeah, that is true as well. Yeah.
Yeah, it was very interesting. It was.
Yeah, but I don't. In my observations, I haven't come across any mechanistic studies investigating that. But you are right. So probably some kind of ECM profile is different that gives the skin its fullness. The fullness is the feature here. As soon as the underlying layer starts becoming frail, the upper layers also starts crumbling, and the fluid carrying capacity of the layers are also significant here.
Dr. Sandeep, hi.
Yes.
Vijay here.
Hi.
Yeah, I think I have two questions. The first one is regarding the molecule that you have put up for your presentation, J147, if I'm not mistaken. I missed few of your slides, so I'm assuming that this can cross the blood-brain barrier.
Yes, it is.
Okay, and does it have a plasma membrane permeability, or does it act via a receptor?
It acts. It doesn't act through receptor. It has the permeability through the plasma membrane, and at the cellular level, it directly acts on the ATP synthase.
So you have.
Hello. Hello.
Hello. So you have a molecule that can. Yeah, I can hear you. So you have a molecule that can directly modulate ATP synthase.
Correct. It directly goes and binds ATP synthase. So that's again, and that goes back to our some interesting link with India as well. It's a modified molecule from curcumin. So it was because, as we all know, curcumin has very bad biological absorption, right? So investigators at the Salk Institute basically modified it, and I would like to call it as a refurbished version of a not curcumin exactly, but the parent molecule. You can say that it has the parent structure of a curcumin.
All right. So it can. So at the organelle level, it doesn't affect any organelle. It just goes and binds one specific enzyme.
It specifically binds with ATP synthase, and the preclinical studies have demonstrated it. And currently, it's going. It's currently performing clinical trials in Alzheimer's patients. The reason why I'm using it is because, to begin with, it interacts with the mitochondria, altering the redox state. And second, and again, it's not the molecule that I synthesized. It's a molecule synthesized by one of my collaborators. So the reason why I approached her to establish the collaboration is by understanding the fact that there is a redox alteration in wound healing. At the same time, if you can target ATP, if you can target mitochondrial components, thereby altering the redox state, you have a likelihood chance of accelerating the healing. So that is the whole premise, and that is the whole flow, the expected mechanistic diagram that I have listed over there. I haven't started working on it. I just started working preliminarily on in vitro models on that as well. I haven't started with the wound healing work yet. And nobody has done wound healing work with that molecule.
Okay, so that actually brings me to my next question. So during wound healing, there is a shift in the phenotype of the macrophages.
Correct.
So do you see, there's a lot of work that is going on regarding this molecular switching. Is it just one switch, or is it like multiple switches that we are talking about? Because if we are going to regulate it, and each diabetic patient, no two diabetic patients, which I believe are the same, how do you think that we can actually tackle them with these inhibitor molecules?
Yeah, there are multiple switches that happens, and the multiple switches happens are at subclass levels. Not every macrophage gets switched. So the switch is mostly from the the switch is mostly from, so the regular switch is from the pro-inflammatory to the anti-inflammatory, right? But that switch doesn't happen in a chronic wound bed because of the tendency of macrophages to linger more towards the pro-inflammatory state, elaborating the pro-inflammatory phase of the healing response. So if you ask me, are there ways to target that particular switching? There are efforts in that direction, but I haven't seen any solid publications out there that through a molecule you can basically skew that switching to more towards an anti-inflammatory phase. There are works that are going in that direction. One of my friends at University of Chicago, Tim Coe, works. He's a very well-renowned macrophage biologist, and he works exclusively in that area regarding macrophage switching in the chronic wound bed.
So this molecular switching, no, that brings me to my next question. This molecular switching that you have talked about, does it actually happen in the wound bed, or does it actually happen elsewhere and they can migrate over there to the wound?
It has two possibilities, right? So it happens in the wound bed. At the same time, that particular macrophage recirculating through other systemic locations and reticular endothelial sites informs the cell population and the reticular endothelial bed, especially the bone marrow compartment, to repopulate that particular skewed one. So both directions it's happening.
Okay. All right. Thank you so much.
So that's why I try to mention that it's a much more vicious cycle. So the switch that yeah, go ahead.
It's a more elaborate process. Actually, it
Yeah,
can't be tamed. It can't be tamed very easily. That's why.
Yeah, it can't be tamed very easily. That's the problem, right? So it can't be tamed very easily, and at the same time, we can't even correctly tame the switching that is happening in the wound bed, right? Because that again is the exact reason why chronicity is existing. But at the same time, if this macrophage starts communicating with the rest of the hematopoietic pool to basically elaborate their clonal expansion, then that becomes a much more elaborate problem to deal with.
Yeah.
Like the epigenetic modification happen related to microbiome, and there is any changes we can actually identify in mitochondrial defect or regeneration process
with the alteration or with the epigenetic changes that is happening with the microbiome.
Yes.
Yeah, it is expected, right? So if you can demonstrate epigenetic changes in the mitochondria,
it will directly affect the mitochondrial membrane fluidity and redox signaling.
Hopefully, right? Because if the epigenetic alterations in the microbiome cellular interaction is some way altering the bioenergetics of the cell, then obviously mitochondria comes to play. Correct?
Yeah.
Yeah. As I have discussed, so most of the cellular interactions has to do with bioenergetics, right? There is some exchange of energy. So in that particular context, you can obviously expect that mitochondrial alterations, mitochondrial dysfunctions, or mitochondrial characteristics are switched with respect to microbiome interaction with various cellular populations. But I'm not sure if you change your gut microbiome, whether that can help with the skin wound healing. I haven't come across any of those studies.
No.
But those are probably the directions the microbiome field is going, right? Because there are a lot of studies with Alzheimer's and GI altered GI profiles, so on and so forth. So, so that probably is the right direction to go.
And specific aging are very much related, skin aging process and.
Yeah, so that's a good question. So whether there are telomere shortening that is already an established mechanism in aging, whether that can be seen in an altered wound characteristics, it's not out there, but I will bet that it's there. So those kind of changes can be easily reflected in the skin population as well, and aging, again, it's a unidirectional path, right? You can't go back. You are all going in one direction. So, and with aging comes a lot of mitochondrial dysfunction as well, right? So when mitochondrial dysfunction comes, then ROS and free radicals and oxidative stress and everything gets messed up in that biological milieu. And aging basically accelerates that, and the DNA characteristics and various other genetic players that drive these are also dysregulated with time.